Hello, my name's Joss Langford. I'm the Chief Technical Officer of Active Insights. Active Insights is a digital healthcare company that specialises in the measurement of behaviour and lifestyle using our own professional wearables. In the talk today, I'm going to discuss some of the aspects of developing digital markers, biomarkers for healthy ageing and public health. In my talk, I'm going to cover 4 topics: defining the data to collect, collecting that data, creating meaningful outputs from that data, and thinking about how we recognise those outputs as biomarkers. Some of the best guidance on clinical biomarkers available at the moment comes from the FDA, and they have a very clear definition that includes biomarkers as an indicator of a normal biological process. And we'd agree that ageing is a normal biological process. It goes on to describe the types of biomarkers that there are and what they measure. And it's very specific to say that a biomarker isn't something that measures how an individual feels, functions, or survives. And that's important from the FDA perspective because these are considered aspects of clinical outcome. But they give us a bit of a challenge because as well as aging being a normal biological process, we would also describe it as a decline in function, or to look at it from the other way from the WHO, as a process of developing and maintaining functional ability that enables well-being in older age. What I'm not trying to say or suggest is that the definitions from the FDA are incorrect, or that we're incorrect to think about biomarkers in the context of healthy aging, but there are some challenges about language that we need to be aware of. The same guidance goes on to define 7 types of clinical biomarkers, and they're shown on the screen here. And it's only the last of these, safety, where public health is actually mentioned. This is a great list, but when you think about it from a public health perspective, we might think that perhaps that there is a category of biomarker, preventive biomarkers, that we should be thinking about adding to this list. So over the last year, some of the thinking that we've been doing as a business in this area, along with many other leading lights from the industry, academia, and from the policy area, has been with the Digital Medicine Society on their Playbook. And this is a fantastic collaborative piece of work. It's over 400 pages of open source content that I would encourage anyone to go and investigate. The links are for this and for all the rest of my references are at the bottom of the slide here. And it introduces a guide to successful remote monitoring in clinical research, clinical care, and in public health. And throughout this, we do talk about biomarkers, but main we talk about measures. And that is part of that is a response to these questions about how we define biomarkers very specifically. Just picking up one page from this, and it's referenced there down at 360. This is one of the first pages of the section on public health, and it talks about the role of digital measures for public health. So 3 streams there, one about supporting decision making and various aspects from that, one around preventative medicine, and a final one at the bottom there about how we prioritize the allocation of resources based on information that comes back from these digital measures. When we think about measures and biomarkers for healthy aging in public health, very specifically, we've got a challenge about thinking about the individual versus the population. So the definition at the top here is one that's taken from Winslow, the science and art of preventing disease, prolonging life and promoting health. Now, when I think about that for one individual, I think about that very differently when I think about that across the whole population. And when we think about the whole population, the context of use becomes very wide. We're treating many different types of individuals, or we're thinking about disease in many types of individuals, and we're looking across many different disease states. Now, when the context of use becomes wide, we've got this huge variance, accuracy decreases, and whether we talk about sensitivity or specificity, this reduces. But because we're talking about so many people, the power can increase, and typically does. So that an accuracy of 60% may not be very useful on a test for an individual, but at a population level, that can be very helpful indeed. And so that good measures for public health may not necessarily be good for clinical trials and vice versa. And for me, one of the challenges that we need to take on in healthy aging is to be able to create measures which are meaningful both to the individual and also at that aggregate level. When we think about what a good biomarker is, the framework that I'd encourage people to use is the V3 framework. And here we talk about verification, clinical validation, and in the middle of those, analytical validation. So the verification stage is about making sure that the sensor devices are doing what they say they do. So from an Active Insights perspective, this is the sort of things that we cover within our medical device dossiers. When we come to analytical validation, then we're starting to work with clinicians, and it may be that we have a step measure which is derived from the wrist, and we want to make sure that that is accurate against a gold standard measure of steps, whatever they are. And then finally, we move into the clinical evaluation, validation, where we're aiming to show, to take the analogy further, that those steps are meaningful for a particular population and in a particular disease state. And this is the best way that we have available to us at the moment to think about what is a good biomarker. And this gives us a context to assess things like accuracy and allows us to evaluate the difference between clinical and public health biomarkers. Dropping a little bit further then into healthy aging itself, and picking up these terms of intrinsic capacity and functional ability that you may have heard of. The WHO puts forward a very strong argument that frailty as a key indicator of aging is much better framed as a positive attribute of capacity and lack of disease. They then go further to introduce an idea of intrinsic capacity, that being the combination of a person's physical and mental capacities. And the reason that's really helpful is that that allows us to think about functional ability. And it allows us to think about functional ability as intrinsic capacity within the environment that the individual inhabits. And it points out that this interaction between the intrinsic capacity and the environment is a dynamic interaction. It is a two-way interaction. And therefore, intrinsic capacity is always going to be best assessed in the community, in the environment in which it's expressed. And we need to then think about this within the community if it's going to be accurate, if it's going to be timely, and if it has the real potential to be preventative. So when we think then about moving measures out of the clinic, we're going from lab-based measurements to free living remote monitoring on a continuous basis. And it may not be with one individual on a continuous basis, but across the population, we need to be constantly sampling that population to be able to maintain good surveillance. We need to be thinking about multiple inputs, objective, subjective, and those community measures about the actual environment, the geographic measures, for example. And the objective measures are here in conjunction with subjective. We're at no point suggesting that objective can overtake subjective. An accelerometer is very good at describing when something happened. It'll give you a good idea about what happened, but it'll never tell you why. And these objective measures are often very beneficial when we want to think about how the subjective measures are subject to bias. We want to move beyond the existing lab measures. We talk about 6-minute walk tests and grip strength tests because they're convenient to measure in the lab, not because they are inherently meaningful in the community. Using objective measures such as accelerometers, we can understand and build proxies to those measures, but we can actually make sure that we use the data from the whole day and capture the individual's entire lived experience. We want to make sure that we're using open format data so that we're creating a future resource for modeling and research. We want to be careful to avoid technology bias. It's very easy when we're deploying technology that we are excluding groups either who don't have the technology or don't have the abilities to access that technology. It's something to be very careful of, and we want to make sure that it's low burden. Low burden for the individual, for citizens. So thinking about no charging, no visits into clinics, mailing stuff, mail back, that it's as non-intrusive as possible, where it can be used long-term, where it's needed on the basis of risk, and that where an individual is using a device at the wear location is appropriate, that it's easy for them to wear. We can also think about reducing the burdens for clinicians and researchers, and here are there are some very good practice that have been developed so that public health researchers don't need to be expert in the deployment of these technologies. And we summarize these around strategy, design and planning, management, creating data summaries, and then interpretation and translation. And activity informatics is a service which has been developed by Active Insights with the University of Exeter for non-commercial clinical trials to provide that expertise into these environments to reduce that burden. I'm going to close out with a case study that brings together some of these aspects in a clinical trial setting that starts to look at some of these wider population elements. And this is a clinical trial called SafeArm, where we are a principal investigator. And it's an international multi-center study, prospective, that aims to develop prediction technologies for patients with implanted cardiac devices. We use wrist-worn accelerometers and mobile phone technologies to follow patients continuously for up to a year. And it's not a suggestion that in all circumstances that we should be trying to do that, but the very fact that we've been able to do that, we've been able to reduce the burden and create processes to be able to do this for over a year, shows the deployability and the feasibility of these approaches at scale. The phone provides an interface for subjective measures coming from the patient reported outcomes if we're in a clinical space, and it gives us the real-time digital connectivity. The accelerometers provide objective measures of physical function and of lifestyle. So some of the thinking that we have about how we use data from the accelerometers there in terms of the types of measurements and biomarkers bring us from the real detail of the individual data through to the population measures. At the base level, we're characterizing sample level data. This is the data straight out of the sensors. This is the verification stage I talked about earlier. Then we use that data to classify behavior and physiological events. So for example, a 5-minute bout of walking. We can then bring those together into short-term summaries. So we can look at activity summaries over the most active few hours of the day, and we can look at immediate trajectories. One of the most common levels is to look at daily summaries. These are very intuitive for clinicians and for patients. And then we can bring together daily summaries over the longer term to look at trajectories. So increases in sleep time that may be positive or negative, changes in people's peak physical activity capabilities. And then at the last stage, we start to look at these at the population level. And this is where we're thinking about how can we use these same measures in the cardiac space in wider population health as indicators of intrinsic capacity. The title of this last slide I've taken from 2 of the papers which are listed at the bottom there, which is their operationalizing intrinsic capacity, and just modified that to stress that what I've been talking about here is digital measures of intrinsic capacity. There are many measures, but I think for all of these, there are 3 key takeouts from what I've discussed. We need to collect open format objective data, and we need to combine that with subjective and community data. And we need to do this in a continuous way, which is low burden to the patient and also scalable. We need to focus on creating a panel of measures that use all the data. So we're not just taking a very small amount of the data that mirrors what we already do in the lab. And that allows us to think beyond intrinsic capacity to functional ability by combining information about the lived environment. And lastly, we need to think very carefully and push forward the validation of digital measures and biomarkers for healthy aging, making sure that they're appropriate for both public health and for clinical use, and that they're suitable at scale and trusted, again, both by clinicians and by patients. Thank you very much.